Chapter 1: Beyond the Typical Medicine Cabinet. Most medicines found in the typical family medicine cabinet are comprised of chemicals that are assembled from other chemicals. However, is a rapidly growing category of medicines known as biologics that are usually more likely to be found in a clinical treatment center or a home refrigerator than in a home medicine cabinet. Biologics represent the newest weapon in the pharmaceutical arsenal. First introduced for human therapeutic use almost 40 years ago, today they are at the forefront of medical disease intervention and are making it possible to address certain illnesses where no other treatments have been successful. But fundamentally, it's the biology of disease that drives the decision of which type of medicine to develop. Chapter 2: Structural Diversity Among Biologics. Biologics are large molecules and often 1,000 times larger than those found in other medicines. They are often proteins, such as monoclonal antibodies, or hormones like insulin, used to treat diabetes. These are very complex and potent medicines that are produced from living systems. While early examples of biologics were based on naturally occurring proteins, more and more unique structures and disease intervention strategies are being introduced every day, adding diversity to the registry of medically approved biologics. Chapter 3: Quality Attributes. Because of their complexity, biologics require much more work to produce and purify, and to test for quality attributes. These quality attributes are measurable properties or characteristics of a drug that define its overall quality. Laboratory test methods are crucial at every stage and in every aspect of biologic development, from screening to characterizing to ensuring appropriate quality for human use. The study of these properties, as well as the development of the tools used to study them, is collectively known as attribute sciences. While some of the test methods in current use have been around for up to 100 years, new analytical methods are continuously being developed. These methods afford greater sensitivity and resolution to the chemical and structural attributes that are designed into the molecule and that are essential for function. In fact, the introduction of some of the most powerful laboratory equipment and methodologies operating today has enabled the development and availability of drugs with greater potency, specificity, and efficacy. Chapter 4: Monitoring and Controlling Biologic Properties. All of the laboratory test methods that go into a drug characterization and control strategy must be demonstrated to meet minimum performance standards. This is done through experiments that interrogate a method's accuracy, precision, reproducibility, repeatability, specificity, and robustness. There are many government agencies and industry oversight bodies, like the U.S. Food and Drug Administration, the International Council for Harmonization, and the United States Pharmacopoeia, that establish laws, guidelines, and standards on how drugs are tested. In general, the properties of a biologic that are monitored and controlled by laboratory methods can be categorized into four main areas: purity, potency, higher order structure, and levels of residual impurities. Each of these areas will be featured in greater detail in companion learning modules in Attribute Sciences. As with most products, biologics are only as good as the raw materials and processes that go into producing them. Raw materials are often subject to the same scrutiny as the products themselves, and will also be discussed in greater detail in this pathway. Chapter 5: Better Science Leads to Better Medicines. A deeper knowledge of disease biology has enabled the strategic selection of the types of medicine needed to address specific biological mechanisms. Along with technological advances in both laboratory sciences and the processes used in the large-scale production of biologics, this is making it possible to deliver more sophisticated medicines with greater quality than ever before. We can now look forward to the elimination of more and more diseases that were previously thought to be untreatable. While there is an urgent need to introduce and deliver new life-saving medicines to patients quickly and affordably, there can be no shortcuts when it comes to ensuring quality, safety, and efficacy. The consequences of undetected errors in the production of any biotherapeutic could be more dangerous than providing no treatment at all.